Past paper questions. Let's start doing the past paper questions of biological molecule. I'm sharing the screen, and these are the past paper questions of February, March, and May, June 2023. I hope you can see the screen.
Yes, ma'am.
So let's start. Let me first do a few questions with you. I'm going to explain the questions and their solutions to you, and then yeah. So let me start. Now you can see this first question. The table shows some steps that can be made in carrying out the Benedict test. Which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution? You know what a semi-quantitative test, in which you can partially estimate the concentration of glucose in a solution, or the concentration of reducing sugar in a solution. This we have discussed in, and we did it in paper 3 as well, that how can we carry out a semi-quantitative test. Let me first explain you what a semi-quantitative test is. Semi-quantitative test is that if I give you, let's suppose, 5 glucose or reducing sugar solutions, and you know their concentrations, they are known, like this one is 10% reducing sugar solution, this one is 30%, this one is 40%, this one is 80%, this one is 90% reducing sugar solution, and you know their concentration. These are the concentrations whose these are the solutions whose concentrations are known, and I ask you to do their Benedict test. You will do the Benedict test and you will record the color changes and make a color chart for your help. That this one has given you green color, this one has given you yellow color, or this one has given you brownish orange, this one gives you orange, this one gives you red color. So you make up a chart for all of these, that this is giving me green color, this is giving me yellow, this is giving me brown, this is giving me orange, this one is giving me red. So you will make a chart out after doing Benedict of all these known concentrations of reducing sugar, or even if I say glucose, but it would be better if I say reducing sugar, because there are 5 reducing sugars. These 5 reducing sugars, you know, are the 3 monosaccharides in the syllabus: glucose, galactose, fructose. All these are reducing sugars. And out of the 3 disaccharides in the syllabus, lactose and maltose are also reducing. So if we are doing Benedict and we are getting a positive result, we never know which reducing sugar is this, whether it's glucose or galactose, fructose, reductose, or maltose. So we cannot say glucose is present. We should have said that reducing sugar. We should say reducing sugar is present. Which one? For this we need to further test that if it's glucose, galactose, because there are further identification tests for all these reducing sugars. And the only non-reducing sugar we know is sucrose. Sorry, sucrose. So the only non-reducing sugar is sucrose, and here we are doing the semi-quantitative test on a reducing sugar. So these are 5 known concentrations of reducing sugars. And if I want to estimate the concentration of this unknown reducing sugar solution, unknown, I do not know what is the concentration of this solution. So this is unknown reducing sugar solution. How would I find it out? I can simply do the Benedict test and can match and compare the color which I get from these 5 colors. Let's suppose I get brown color, or I get brownish orange color. So, if I get brownish orange color of this unknown concentration of reducing sugar, then I can say that my unknown concentration lies somewhere between these two, 40 and 80. Let's make it 60 because 40 would be very close and it won't give brown. So let's say I will say that my unknown concentration is giving me brownish orange color. So that lies somewhere between this brown and orange, and its concentration lies somewhere between 60 and 80. So it can be, I can just estimate, I'm not sure, I can simply estimate that it is about 70% reducing sugar solution. That is called as a semi-quantitative test, that partially I have estimated the concentration of my unknown reducing sugar solution with the help of some known concentrations. I did their Benedict test and made a color chart out of them, and then I compared the color of my unknown reducing sugar solution with the color chart, and I realized it's somewhere between brown and orange. So that is almost not 100%. I'm sure it can be 72, it can be 76, it can be 79 even. So I can estimate roughly and partially that it's 70%. This is called semi-quantitative test. Is it clear to you? Is it clear what a semi-quantitative test is? Is this clear? Can you hear me? Is it clear with it? Am I audible to you?
ना आपकी आवाज थोड़ी कट रही है.
Um, I can hear you very well. I'm surprised that did you get it what I say?
Yes मैम, बट आखिर में थोड़ी कट रही थी आपकी आवाज.
Okay, let me repeat it. I have said that semi-quantitative test is a test which can help me in roughly estimating the concentration of an unknown reducing sugar solution. I have given you, let's suppose I'm giving you these 5 concentrations, their reducing sugar concentrations are known. 10%, 30%, 60%, 80%, 90%. Am I audible to you?
Yes ma'am.
Okay, so these are known concentration, and I have done their Benedict test, and I have made a color chart, which the color of the colors which I'm getting from these solutions after doing the Benedict test. I'm getting 10%. I'm getting green color, 30% for 30% yellow, for 60% brown, for 80% orange, for 90% red. So I'm going to make a color chart in front of their percentage concentration. I'll write the colors, then I'll hold the unknown reducing sugar solution, and I will do the Benedict of this, and I will observe the color. Let's suppose I get a brownish orange color. The brownish orange color will direct me to between brown and orange, these two test tubes. One is 60%, other is 80%. So I will roughly estimate that my unknown concentration lies somewhere between this brown and orange, that is brownish orange. So it means that would be almost or about 70%. So that is my rough estimation. Na? I'm not sure if it's 70% or 74% or 78% or 76%. I can roughly estimate it is 70%. This is called a semi-quantitative test. Is this thing clear to you?
Yes ma'am.
Okay, so now the question says that which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution. So what are the steps? You can see standardized volume of Benedict solution and a volume of test solution. That is, of course, we are going to standardize. It is correct. Standardize to karenge na, we should keep them same. Then boil with hydrochloric acid and then neutralize with alkali. That we are also no, that we are not going to do. Why should we do this? That's for the non-reducing sugar. This is the condition for the non-reducing sugar, and we are doing the test for the reducing sugar. So why should we do this boiling with hydrochloric acid and neutralizing with alkali? We are not doing it for non-reducing sugar. We are not doing it for sucrose. So we won't do it. So this is correct. This is correct. This is correct. But of course, this one is incorrect. So we won't go with this. We can go with A and C both. Now let's see the final column. Standardize boiling time with Benedict solution and compare final color with numbered color standards. That is correct. We are going to do this. We are going to standardize boiling time. We are going to boil the solution for a certain period of time that should be same for all of these. And as soon as we get a final color, which should compare our color with the numbered color standards with the color chart which we have made. We're going to compare it. So this option is correct and this wrong which is written is wrong, because we will do this. This is correct and this is also correct. And these two are giving the correct for this these two columns. So our answer is C. You understood it or any problem in it?
No, ma'am.
Okay. Now the next question says, I'm erasing some portions. Now the next question simply says, the diagram shows three examples of different bonds. Can you identify these bonds? This between hydrogen and oxygen, which bond would it be? This is hydrogen. That oxygen is partially negative, that hydrogen is partially positive, and the force of attraction between the two is hydrogen bond. This is a peptide bond. That is one amino acid over here, other is over here, and that bond is peptide bond. And this is between S and S, disulfide linkage. So which bond holds the secondary structure of proteins together? What holds the secondary structure of proteins together? In the primary structure, we know it's peptide bond. And to become a secondary structure, alpha helices, beta pleats, hydrogen bond comes in between. So the one which holds the secondary structure is the hydrogen bond, and that is one only, bond one. Thirdly, insulin is a globular protein involved in cell signaling. It is transported in blood plasma from the cells that synthesize it to target cells. A molecule of insulin has 6 sulfur containing amino acids. What are sulfur containing amino acids? These are cysteine. Sulfur containing amino acids, they have in their R chain, they have H and S, and in their R chain they have H and S. And what happens? This S and S makes bond and hydrogens are removed. This makes bond, that is disulfide linkage. Hydrogen is removed from both sides as 2 protons, and bond is formed between S and S, and that is called as disulfide linkage. You can see it over here. This is the disulfide linkage. Yes. This is one amino acid, this is other, and in their R chains they have S and H. H is removed and S and S have bonded. That's disulfide linkage, which they are talking about over here. So here you can see 2 protons have left and disulfide linkage is being formed. So you can see which they are saying that a molecule of insulin containing 6 sulfur containing amino acids means they have 6 sulfur containing amino acids means they have 6 cysteine and has 2 polypeptide chains. Which statements about insulin are correct? 6 sulfur containing amino acids means 6 cysteine honge. If there are 6 cysteine, so among 6 cysteine, if this is one cysteine, this is other, one disulfide linkage is being formed. So if there are 6 cysteines, 3 disulfide linkages would be there, because between 2 cysteines there would be one disulfide linkage. So between 6 cysteines, 3 disulfide linkages will be formed. So let's see which one is correct. An insulin molecule has a quaternary structure. It has a tertiary structure because it has a quaternary structure. That's correct, because it has 2 polypeptide chains. That is correct. Then insulin polypeptides are held together by 6 disulfide bonds. That's incorrect, because I've told you if there are 6 sulfur containing amino acids, means 6 cysteines. 6 cysteines are when bonded, one disulfide, 2 cysteines are bonded to make one disulfide. So 6 cysteines are bonded to make 3 disulfide. 6 disulfide bonds is absolutely wrong. That's incorrect. Thirdly, amino acids with hydrophobic R groups would be found in the center of an insulin molecule. It is correct, because it's a globular protein, and the protein becomes globular when all the hydrophobics are in the center and all the hydrophilics surround the hydrophobics in the at the corner and at the edges, so that the hydrophilic centers can interact with the polar environments and all the hydrophobics are all compact in the center. So this is also correct, and 1 and 3 option, that is C, is correct. Now the next one. Which statement correctly explains why blood plasma can be maintained at a stable temperature? Blood plasma can be maintained at a stable temperature. You know that blood has a high specific heat. It has a high specific heat capacity, which is that in order to raise the temperature of 1 gram of water only to 1 degree Celsius, lot of energy, a large amount of energy is required. So that is high specific heat capacity, and that is why if the external temperature changes, a blood plasma, which is 80 to 90% water, its temperature does not change very quickly. So that is why the blood plasma is maintained at a stable temperature, and enzymes and hormones can keep on working easily within blood plasma, which is made up of 80 to 90% water, and the water has high specific heat capacity. So the external temperature changes, water's temperature does not change quickly, and it is maintained at a stable environment. So B is correct. Are you understanding it? Do you understand the concepts?
Can you repeat it, please?
Okay. Now, question number 5. Which set of steps in the is the best method for conducting the immersion test for liquids? You know, for immersion test, what do? We mix up the sample and ethanol, and then we add water to it to see the cloudy emulsion. So, add 2 centimeter cube of water to the sample. Pour the water into test tube containing ethanol. Lipids are present if mixture becomes cloudy. That is wrong, because you have added 2 centimeter of water to the sample, and when you're adding first you're adding water and afterwards you're adding ethanol, so that's wrong. Then add 2 centimeter cube of ethanol to sample and shake. Pour the ethanol into test tube containing 2 centimeter cube of water and boil. That boiling is wrong. We never boil in emulsion test. That is also then wrong. So the only option left is D, and that is correct even. Add 2 centimeter cube of ethanol to sample, shake it well. Then pour the ethanol into a test tube containing water and shake again. Lipids are present if mixture becomes cloudy. So there is no heating, no boiling, and first water sample and ethanol are mixed and then water is added. So this D is correct. That's the right way of doing ethanol emulsion test. Now in the next question, you can see a student was provided with a solution of carbohydrate. They removed 2 samples from the solution and performed test on each sample. You can see this is the carbohydrate solution, and he is sample 1, sample 2. In sample 1, he has added Benedict, and the solution remained blue. So that is simply the test for reducing sugar. He did it, and he got a negative result. That is the test for reducing sugar. And this one is the test for non-reducing sugars, that is sucrose. Reducing sugars, I have told you, are glucose, galactose. By now you should be knowing it. Fructose, lactose, maltose. And if we talk about this is the test for non-reducing sugar, and non-reducing sugar is sucrose. And that is only sucrose. So you can see in this second sample, we have boiled it with hydrochloric acid and then we have neutralized it using sodium hydroxide and then Benedict acid. When we add Benedict acid, then the solution turns blue. It means that it is giving you, not only it's a non-reducing sugar test, it is giving you a positive result because it does not remain blue, it has turned to be yellow. So it is giving you a positive result, turning yellow. It means non-reducing sugar, sucrose is present. So let's see the options. What explain which statement? Let me enlarge it. I zoom it in just to show it to you. Zoomed out. Acha. Which statement explains the result? Condensation reactions occur in sample 2 to release reducing sugars. Glycosidic bond in a polysaccharide have been broken to release reducing sugar. Glycosidic bond in a polysaccharide have been broken to release reducing sugar. Yes, because you know when we do this acid treatment and we neutralize it with sodium hydroxide, neutralization is because Benedict doesn't work in acidic condition. But when we do this boiling, it breaks glycosidic bonds and as a result releases reducing sugars. So when glycosidic bonds are broken, reducing sugars are released, which gives us a positive test for with Benedict in the form of a yellow color. So this is the correct option. Condensation reaction takes place when two monosaccharides join to make up a disaccharide, and here nothing is being joining. It's a catabolic reaction in which the polysaccharides are broken at this level because of the HCl. So यहाँ पर condensation नहीं हो रही, यहाँ पर hydrolysis हो रहा है. So if it would have been hydrolysis correct हो जाता, लेकिन condensation कह रहे हैं जिसमें चीजें join होती हैं, break नहीं होती. यहाँ पर आपकी glycosidic bond break हो रही है और reducing sugar आपकी release हो रही है जो कि आपको yellow color दे रहा है. So B is the correct option. Sample 1 shows that sucrose is present. That is wrong. Sample 1 shows that neither sucrose nor that reducing sugars are absent. ये बता रहे हैं. The change in color to a yellow solution shows that glucose is present. No, it shows that sucrose is present. So that is also wrong. They are all wrong. Now, if we talk about the next, which molecule contain at least two double bonds? You know, hemoglobin and collagen are made up of amino acid, and remember, this is the structure of an amino acid: R chain, carbon in the center, H over here, and C, and either N and H, double bond O and OH. So this is one amino acid. When the amino acids make bond with another amino acid, one H is removed from over here and OH is removed from here. As a result, water is released, condensation reaction takes place, and this carbon and N of the other is joined this way. Let me draw it. R, H, H, and C double bond O, and in the next amino acid H, R, C double bond O, OH. So in this next amino acid we have N, and this one of the H, as I told you, is removed and the other H is left. So this is the peptide bond between the two amino acids. This is peptide bond. We did it before as well. And you can see that within the two amino acids, there are two double bonds. One is this, other is here. So there are two double bonds. So there are two double bonds. One is this, other is the second double bond. So you can see that hemoglobin, collagen are huge proteinaceous structure made up of so many amino acids. So if within two amino acids there are two double bonds, so between hemoglobin and collagen, when there are so many amino acids, there will be a lot of double bonds. So these two are obviously correct. And if you talk about sucrose is a simple structure made up of, you know, one glucose and other fructose. So glucose and fructose make up sucrose, and there is no double bond over here. So sucrose is does not contain double bond, but they have a lot of double bonds. So that the correct option is B. Next is what describes cellulose? Cellulose, you know, is made up of beta glucose. I have told you that it is made up of beta glucose, and every alternative beta glucose turns upside down at an angle of 180. When I taught you about cellulose, I taught you this thing. So that's why it is made of beta glucose, and the bond between the two beta glucose is 1,4, because this is carbon number 1, this is carbon number 4. So that is 1,4 glycosidic bond. See, this is one beta glucose, this is the other beta glucose. This second beta glucose had to turn upside down because its hydroxyl was not right in front of this carbon number 1, that it was on the up, it was on the in this over here hydroxyl in case of beta glucose was above the chain. That is why in order to bring it, it was down the chain, it was over here. So that is why, you know, it took a twist and of 180-degree angle upside down, so that the two hydroxyls have brought closer, and as a result the bond has been made. Because in case of beta glucose, you know, let me explain it to you in detail. In case of beta glucose, you know, carbon number 1's hydroxyl is above the ring. So if we have two beta glucose, and in case of both, hydroxyl is above the ring of carbon number 1, but carbon number 4's hydroxyl is below the ring. So if the bond will be formed in this way, it will be a weak bond. In order to make it a strong bond, this second beta glucose rotates upside down. When it rotates upside down, all the things which are above the all the things which are above the ring go down, and all the things which are below the ring move up. So as a result of this, this will remain in its position, but the second beta glucose, which has turned upside down, its hydroxyl over here will go up, and its hydroxyl over here, which was above, here it was down, now it is above. And here this carbon number 1's hydroxyl was above the chain, now it is down the chain. So now the bond formation is very easy. From here hydrogen, and from there hydroxyl will be removed. Condensation reaction will take place, water will be formed, and this O is now in between, and this is how the bond is formed. You can see first beta glucose, and this is the second beta glucose, which took a twist of upside 180-degree angle upside down. So a bond is formed above the chain. So this is how beta glucose is, and carbon number 1 and 4 are making up the bond. So the glycosidic bond is formed, and this is said to be no, it's not a branch chain. It's an unreactive linear chain of 1,4 beta glucose. There are no branches in cellulose. Cellulose is without branches. It is unbranched. It has 1,4 glycosidic linkage. It is linear. So that is the correct description. 1,4 beta glucose, linear chain, unreactive because it does not have branches. So it is not very reactive. Is this thing clear to you?
Yes, ma'am.
Okay, so now I have another class, so I will continue with the rest of the MCQs starting from this ninth MCQ in the next class. I hope these first 8 MCQs are clear to you.
Yes, ma'am.
Are these all clear? If you will revise the topic, they will be more clear to you, and you will understand them more clearly if you will revise the topic more, and we will keep on doing the past paper to make your concepts more clear. Okay? Thank you very much. Allah Hafiz.
Allah Hafiz, ma'am.
Yes.